Alright, in the last video I did a terrible recap of the physics unit on energy, magnets, and using electricity. And then I lied at the end of it because I said I was going to talk about, I think, DNA or genetics and astronomy. But I don't need to talk about genetics and astronomy because from genetics on, those summaries are on videos. And so you can easily go back and dig up those videos from the lessons if you need to recap them. So we're only going to look at DNA. And then we are going to say that's enough, except some of you are going to learn to watch the videos. It's become apparent that some of you don't watch the videos, and then you struggle with the worksheets. What a surprise. Okay, DNA summary. So we were interested in the structure of DNA, and so we wanted to have a good command of that, and then we were able to talk about how we can use DNA to ultimately make protein. So you want to know why DNA is important. That's what it talks about here. It contains the genetic information. It's important that genetic information is passed on properly from one generation to the next. And last year you learned about mitosis and meiosis to get some sort of sense of that. And so you should know what genes are, the short sections of the DNA that basically are going to code instructions. You should know that DNA is the double helix or twisted ladder. You should know that a nucleotide is a sugar, a phosphate, and a nitrogenous base. And those nitrogenous bases are going to be our A, T, C, or G when we're talking about DNA. And even though there's just 4 nitrogenous bases, we get a lot of different sequences. And that means we can code for a lot of different proteins and traits. We have to remember that A and T, adenine and thymine, fit together, and C and G, cytosine and guanine, go together, and we call them complementary. So DNA replication. DNA for a small thing is a big thing. It's like a jumbo shrimp. It is too big to leave the nucleus. And so DNA can replicate. That means it can make a copy of itself. And that has to happen in the nucleus since that's where it's trapped. So the DNA will unzip, and then you'll have nucleotides floating around, and they will just come in and lock in to nitrogenous bases that need a pair. And then it just simply zips back up. And I'm making it sound very easy. If you go on to senior biology, they give you a lot more detail. But that's really an overview of what happens in replication. Protein synthesis involves a number of major steps. So what we're trying to do is we're trying to take information from the nucleus and the DNA, and we're trying to take it into the ribosomes that are outside the nucleus in the cytoplasm. And the ribosomes are where you're going to be making your proteins. Then you say, well, what on earth are proteins? Well, proteins are made up of smaller units called amino acids. So let's say you had 9 blocks of Lego and you stitched them together. Well, each block of Lego would be like an amino acid, and the 9 blocks stitched together would be like a protein. So in transcription, what happens is messenger RNA, and remember it is just a single strand, not a double strand, will come in. The DNA will unzip, and the messenger RNA will fill in. The nucleotides fill in properly. But now, in the RNA, we have uracil with a U because there's no thymine in RNA. The messenger RNA, once it's made that copy of a section of the DNA, it can leave the nucleus and fit through the nuclear membrane and get to the cytoplasm. And the DNA will zip back together. Now the messenger RNA has arrived in the cytoplasm and it heads to the ribosomes. Once it gets to ribosome, translation can take place. And the messenger RNA is going to be a long strand, but it gets read in 3 unit sections. And what happens as it goes through the ribosome, on the other side will be a complementary nucleotide. And so the messenger RNA has what we call codons, triplet codons, because 3 letters. And the transfer RNA has the anticodon. It has the 3 letters or bases. And they match up. So the transfer RNA comes floating in, and attached to transfer RNA, so at one end you have the bases, at the other end you have the corresponding amino acid. And that's where you use the codon chart, because once you know what the codons are for the messenger RNA, you can just go boom, 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 all the way along the line and figure out the sequencing of the amino acids. And once they're joined together, you've made a protein, and another word for protein is polypeptide. Poly means more than one, and the bond that forms between amino acids is called a peptide bond. So that's where that's coming from. You should know differences between DNA and RNA, and there are certainly some fundamental ones. One, of course, are the 4 bases. They differ in that you have a T in DNA and a U in RNA. The shape or structure varies because DNA is a double helix and RNA is going to be a single strand. DNA is bigger, trapped in the nucleus. RNA is small enough to move in and out of the nucleus. You should know about what different proteins are in terms of their functions, and that was provided in the summary. And then you had a test, and many of you found that test to be straightforward, and you knew how to use the codons chart. So that's our recap of DNA. That's sort of our recap of the course, because you can go hunting for the other summaries, and the other summaries should be organized in your binder. And also, the other summaries are also available in video form on different lesson days if you've been looking at them carefully. Many of you would do fine on the final. The astronomy section would be brutal because for some reason I just have nasty questions there. The chemistry section you should dominate because it's so recent. All right, so that's our recap. I'm not going to give you a big package of work to prepare for the final since there is no final. But if you said, you know, I want to take this seriously, what you would do is probably look for all the different practice tests that you've had throughout this course and see if you could answer those questions without assistance. And if you could do those, you have a good idea that you probably do fine on a final exam if we had one. All right, so for the rest of the week, we are going to do some other weird and strange stuff. And there might be the odd other assignment, but the course is now really winding down. So if you've been working hard, you deserve a break. And if you haven't been working hard, you better have an intense week and get stuff in.